Alright class, so let's do a sample one. Okay, so this is all hand drawn. Sorry, please forgive me for this awful drawing, but this is the best I could do on my screen. Okay, so I will ask you to count the number of cells in that you see. Okay, so let's get started. Are you counting this one? No. Are you counting this one? No. Are you counting the ones that are touching the middle? Yes. 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 Oops. Not this one. Not this one. Are you counting this? Yes. 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 And that's it. So how many cells are here? Let's count. 1, 2, 3, 4, 5. No. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. The right answer is 16. Okay, so something like this, you must know the rule. You must know the rule. All right, so after you count, let's say, so I just showed you counting, how to count one box, right? So you will count all 5 boxes, and let's say you have 16 in the first box, then you have 10 in the second, you have 12 in the third, 8 in the fourth, and then 10 more in the fifth. So that adds up to 18, 28, 40, 56. 56 cells altogether. Now, how do you determine the number of cells per cubic millimeter of blood, right? So, this is what we do. You take 56 that you counted in all 5 boxes, and then you multiply this by 100,000. So, what number do you get? 56. Right? These many cells. Now, how do you express this? You express this as 5.6 into 10 to the power 6 cells per microliter per cubic millimeter of blood. Okay? So, this is how we end up writing the actual cell number. So on your exam or on your quizzes or practical exam, whatever, you will be asked to calculate. I can tell you, okay, these are the 5 boxes and these are the number that you counted in each box. How many cells do you see? Tell me the number of cells per cubic millimeter of blood. So your answer should follow this. Now, the number of RBC count varies a little bit between males and females. Males have a little higher RBC count, 4.2 times 10 to the power 6 to 6.9 times 10 to the power 6. However, females have a slightly lower number. All right? Alrighty. So now moving on to cleanup. Okay, so what goes in the biohazard sharp waste? You should know. What goes in the biohazard soft waste? You should know. And what goes in the regular trash? You must know. Based on this information, so right now we have few informations, right? We have hematocrit value. We have our hemoglobinometer count, which tells us the amount of hemoglobin. And also we have, what was the last thing we did? Hemocytometer. We have hemocytometer count. So we have these 3 informations. Based on these, we can calculate 2 things. MCHC and MCV. These are the two things we will calculate. Now let's take a look. The first one is MCHC, also known as mean corpuscular hemoglobin concentration. And this is defined as the average concentration of hemoglobin is a given volume of packed RBC. So let's say you look at this volume of packed RBC here, and by MCHC you can determine the concentration of hemoglobin in this volume. So for this, first you need to know two things. You need to know the hemoglobin concentration that you have from the hemoglobinometer, and remember that is in grams per deciliter. And also you need your hematocrit value. Using these two numbers, you can find out MCHC. So let's do the math. So let's say your hemoglobin concentration, number one here, is 15 grams per deciliter. And let's say your hematocrit, which is the second thing we should look at, is 50%. So these two will be given to you, okay? And you on a test or on an exam, you will be asked, calculate MCHC. So what you will do is, you will take the hemoglobin concentration, which is 15 in this case, multiply it by 100, and then divide it by the hematocrit. So that gives you a 30%. So the mean corpuscular hemoglobin concentration in this case is 30%. Hopefully that makes sense. The last thing that we will talk about today is mean corpuscular volume. And this is defined as the average mass of hemoglobin per RBC in a sample of blood. It's talking about mass of hemoglobin per RBC. Per RBC, okay, per RBC. Okay, so for this, you definitely need the hematocrit value. That's something that you need. And remember, hematocrit is just percentage. Man, it's just percentage. And also, you need your RBC count. Remember the RBC count from the glass slide? So you need, so in this case, it's 4.0 times 10 to the power 6. So with these two numbers, and let's say hematocrit is 50%. You need these two numbers, and the way you calculate MCV is take the hematocrit value, which is 50, multiply it with 10, and then divide it by this number. Sorry, not the whole thing. Divide it by this number. So you put here in the denomination, and you get 125 micrometer cube. Don't forget the units. The units should be, actually you will get multiple choice, so not too bad. Okay, so again, one more time, you need the hematocrit value and you need the RBC count. You take the hematocrit value, multiply it with 10, and then divide it by the whole number of the RBC count. So another example would be, let's say hematocrit value is 45% and RBC count is, let's say, 5.0 times 10 to the power 6. Given these two information, can you calculate MCV? Yes, you can. So in this case, MCV would be 45 times 10 divided by 5. So that would be 90 micrometer per cube. Does it make sense? All right, class. Okay. So that concludes my lecture today. I have also posted two very detailed video talking about blood typing. Blood typing is big, and you can expect harder questions. You can expect critical thinking based questions on blood typing. So if you are confused at any point, please do email me and we can go from there. Okay, so I recommend you watch these videos and then do the work on the post lab. All right, okay class, bye.